Here's the workbook, in fact. It is presented by Dr. Huda Baraco, Kingston, Piruat, Lopez Barbero, and Dahlia from the University of Texas Health Science Center of San Antonio and Laboratory for Biomedical Investigations in the University of Montreal, in Sevilla, Spain.
Good afternoon, everybody. I'm Kalyan, and the topic that I'm going to present is the inactivation of succinate dehydrogenase is sufficient to reduce the Warburg effect. Normal cells generally they depend upon oxygen for their energy requirements, which is termed as oxidative phosphorylation, and only resort to glycolysis in the absence of oxygen, whereas cancer cells, in contrast, depend upon glycolysis for their energy requirements, even in the presence of oxygen. This phenomenon was described by Otto Warburg almost 70 years ago, and this phenomenon is called as aerobic glycolysis or as the Warburg effect. First, the Warburg effect reflects increased glycolysis coupled with reduced oxidative phosphorylation. The end product of aerobic glycolysis is the conversion of pyruvate to lactate, which is mediated by the lactate dehydrogenase gene. For many years, the metabolic profile of cancers has been considered a consequence of this malignant transformation. But now there is sufficient evidence to suggest that the Warburg effect is in fact proved as a selective advantage for the cancer survival. The mechanisms of Warburg are still unknown, and understanding them will be relevant for the study of cancers in general. So we are trying to study this Warburg effect using the cancer that is called as pheochromocytoma as a model system. The pheochromocytomas are the tumors that are derived from the chromatin cells which are nucleus in origin. These tumors can originate from the adrenal glands or from the autonomic tissue outside the adrenals, where they are known as the paragangliomas. They secrete excessive catecholamines, which frequently lead to sustained or paroxystic hypertension, but can as their main clinical features. These tumors are usually sporadic and benign, but can also occur in malignant and are quite indistinguishable from the benign tumors. Mutations in one of the 6 independent genes are responsible for about 30% of the pheochromocytomas, and among these susceptible genes, one of them are the subunits of the succinate dehydrogenase complex, which are the main focus of our study. So the succinate dehydrogenase complex, it's an inner mitochondrial, it's bound to the inner mitochondrial matrix. It's the only enzyme that has 2 functions. It functions both in the TCA cycle as well as the electron transport chain. In the TCA cycle, it functions in the conversion of succinate to fumarate, whereas in the electron transport chain, it helps in the transfer of electrons to ubiquinol. The mutations in B, C, and D complexes of the succinate dehydrogenase have been known to cause pheochromocytoma. Of the 4 subunits, the SDHA is the regulatory subunit, SDHB is responsible for electron transfer, and C and D function in the binding of the heme to the beta catalytic domain. So the big question that we are trying to answer is, does this loss of succinate dehydrogenase function recreate the features that will induce the Warburg effect, and is it a good model to study the link between energy metabolism dysfunction and tumor cellular growth? For this, we designed a study which consists of 3 different models. One is the in situ model, in which we collect human pheochromocytomas from patients with and without SGH mutations and do a gene expression profiling of them. And then we do it on the in vivo murine SDHD knockout mice. In the homozygous mice, it has been proven that SGH mutations are lethal for them, whereas the heterozygous